pleasant afternoon to each and everyone. I am Ma'am Rosie and I will be your science teacher. For this afternoon, we're going to learn additional science concept. But before that, let's try the following. Okay, who are familiar with these love teams? Anyone? You're right. These love teams are one of the phenomenal love teams as of this present time. The first picture are the Katniel. The second one are Lisken. And the third one are Jadine. What do you think the reason behind they are popular? If we take a look on their physical appearance, they are very attractive. What do you think? They inherit those characteristics. They inherited those characteristics from their ancestor, from their parents or even their relatives or grandmothers or fathers. Are you aware with the word blood relatives? We are very confused with these terms or these phrases in such a way that we are related with our relatives not by blood. Take note, not by blood. But we are relatives because we shared common characteristics that are coded on our genes. You will learn further what I am talking about as we go through with this lesson. Okay, our topic for this afternoon is all about codominance, incomplete dominance, and multiple alleles, or what we called non-Mendelian genetics. In our grade 8, we learn about Mendelian genetics. In our grade 9, we are going to talk about non-Mendelian genetics. We will be guided with the following objectives. The first one is explain some principles of non-Mendelian pattern of inheritance using diagrams. Next, illustrate by means of Punnett square across involving incomplete dominance, codominance, and multiple alleles. And lastly, cite the effects of incomplete dominance, codominance, and multiple alleles on a certain organism. As we go through our lesson, we encountered different words. So at first, in order to be familiarized with those terms, we're going to define it. First, genes. When we say genes, it is a unit factor of heredity. Next, alleles. Alleles is a different form of a gene that control a certain traits. Example, capital G and G indicates the seed color yellow. Small letter G and G indicate seed color green. So when we say capital G and small g are both alleles coded for colors, but it carries different characteristics. Next, gamete. When we say gamete, it is a reproductive cell that unite during sexual reproduction. Next, phenotype. When we say phenotype, it is a physical appearance or a trait. Take note, when we say phenotype, it is a physical appearance of a trait. Genotype. When we say genotype, it is a genetic makeup of a certain individual. Next term is homozygous. When we say homozygous, from the word itself homo, which means one, it made up of identical gene. When we say heterozygous, it is made up of dissimilar gene. Next is dominant. When we say dominant, alleles that is always present as characteristic. When we say recessive, alleles that are hidden by dominant alleles. Okay, for example, here's a sample problem. When a pure red roses and a pure... Okay, again, here's a sample problem. When a pure red roses and a pure white roses are crossed, what is the possible offspring? What do you think, class? Okay? Take note, pure red and pure white roses. Okay, take a look at this. So, we are going to use key in order to solve with the possible offspring. Big letter R indicates for the color red. WW indicate for the color white. RW indicate for the color pink. Okay, using the Punnett square. So, class Punnett square is a method used in order to identify characteristics or possible characteristic or possible offspring of a certain organism. Punnett square is like multiplication. It's like using, what do you call that? Table of multiplication. Okay, where do we usually use multiplication table? You're right. It is used in mathematics. Simply solving mathematical formula involving number or numbers. Okay, let's back. Using Punnett square, we're going to cross multiply. So on the upper, we're going to put the key terms on red color, the RR. While on the other side, we're going to put the white color, since the offspring that are going to breed are pure red and pure white. Okay, W times R, or R times W, is equals to RW. R times W is equals to RW. Okay, on the next column, R times W is equals to RW. R times W is equals to RW. So, what is the possible offspring upon breeding pure red roses and white red roses? You're right. The possible offspring of these two variety of roses is pink. or it is indicated on the RW. Indicate for pink. Okay. Those sample problem is an example of incomplete dominance. So, when we say incomplete dominance, it is a pattern of gene expression in which the phenotype of a heterozygous individual is intermediate between those of the parents. So, if we notice, the offspring is pink, which is if we combined red and white, or if we mix red and white, the possible color that arise is pink. So, it is intermediate characteristic between the parents. As of this present time, we are all aware that many people are engaging in planting different kinds of plants, or what we call plantita and plantito. And in our high school days, or elementary days, or college days, we usually engage in this kind of things. What specific subject are we going to integrate? You're right, TLE, specifically agriculture. So in agriculture, we plant different variety of crops. We breed or crossbreed a certain kind of plants in order to get the possible offspring, or the offspring that we want to have. Okay, aside from roses, we can also breed snapdragons and carnation. So those are flowering plants. So example siya ng incomplete dominance. Next. So we are done with incomplete dominance. Next sample problem that we're going to solve is when a red-furred cattle and a white-furred cattle are crossed, what are the possible offspring? So same with what we do on the problem that we encountered on incomplete dominance. So we are going to use Punnett square. Okay, take note the organisms that we are going to breed is red-furred cattle and white-furred cattle. So same breed of cattle, but then different colors. Okay, again, using Punnett square, we're going to assign key for us to solve properly the problems. Okay, we're going to use big letter R for red-furred cattle, big W for white-furred cattle, and RW indicated for red with white hair, or what we call roan cattle. Okay, using Punnett square, same process. R times W is equals to RW. R times W is equals to RW. Next, RW times W is equals to RW. R times W is equals to RW. So if we observe, the results are 100% red with white hair, or what we call roan cattle. So based on the picture, you're going to see a cattle comprises of red with white blotches, or what we call roan cattle. So do you think it is the same with It is the same with incomplete dominance. Think about it. Okay. The example indicated is an example of what we call codominance. Okay, when we say codominance, it is a condition in which both alleles in a heterozygous organism are expressed. So, it only means that both alleles contribute to the physical characteristic of that organism, or what we call phenotype characteristic of that organism. So, those alleles don't blend. Take note, they do not blend, but rather they are both present in the offspring. So, aside from cattle, we observed this on other organisms. For example, on chickens, on horses, on cows, on clovers. So take note, codominance don't blend, but then they are both present in the offspring. So that is the difference from incomplete dominance. In incomplete dominance, they are both blending. In codominance, they don't blend, but then they are both present. I hope it is clear to you. Okay. Next example is an ABO blood type or human blood type. Human blood type comprises of 3 alleles. Take note, human blood type comprises of 3 alleles. So it is written in letter I, small letter A, naka-raise siya sa A, and then small letter I, raised to B, small letter B, and small letter I. Take note, those I letters with A and B are codominant alleles. And they are both dominant to the alleles I. Okay, take a look on the table presented. If you observe, it has genotype and it has blood type. Okay, when the genotype is I raised to A and I raised to A, the blood type of a person is blood type A. If A and small letter I, same with the blood type is letter A. When the genotype is I raised to B, I raised to B, and then I raised to B and small letter I, the blood type of that person is B. And lastly, when I raised to A and B raised to A and small letter I, small letter I, the blood type is O, while if the I raised to A and B, the blood type is AB. So if we notice how many blood type? 1, 2, 3, 4. So technically we have 4 blood type. And how many genotype? 1, 2, 3, 4, 5, 6. 4 blood type with 6 genotype. Okay, donor-recipient compatibility. So, where do we usually use this process? So, we use this in healthcare, in hospitals, or we study this specifically in health mapping. So, it is very crucial or very important that we know what kind of blood type are we going to transfuse on that person? Because wrong blood transfusion result to death. Or even, what we call this, complication for that patient. Okay, on the table indicated, the donor on the left side, the upper side is the recipient. Okay, if the donor is blood type A, the recipients are, so by the way, the legend red, it means safe transfusion, while the, what color is this? Parang fuchsia. Sorry. What color is that? Light pink? Something like that. Okay. But let's focus on the color red one. When we say, when the color is red, it means that the transfusion is safe. So donor type A, the recipients of this are blood type A and blood type AB. When the donor type is B, it is safe to transfuse on blood type B and blood type AB. When the donor is AB, it means that it is safe to transfuse on blood type AB. So if we notice AB and AB only, while when the blood type is O, they are all safe to transfuse on the 4 types of blood. So blood type A, blood type B, AB, and O. So blood type O is what we call universal donor. So based on the table, it is safe to transfuse on different kinds of blood. But blood type AB is what we call royal blood. So universal recipient lang sila, pero hindi sila universal donor. Okay, when we say multiple alleles, it is a gene that is controlled by more than 2 alleles. So based from the example, a very good example of this is the ABO blood type in human, because how many kinds of alleles are present? So there are 3 kinds of alleles present. Okay, let's try a certain activity using what we learned from the 3 concepts presented. Okay, given the problem 1, a cross is made between a red rose plant and a pink rose plant. What is the possible genotypic percentage and phenotype of the offspring? So same process, we're going to use key in order for us to easily solve the problem using Punnett square. So double big R indicate for red rose, double big W indicate for white rose. Okay, using Punnett square. So what do you observe? R times R will result to double big R times R, R times W, RW, R times W, RW. So the genotypic percentage of this is 50% and 50%. So it means 50% red rose and 50% pink rose. So when we asked what kind of genotype present? Or what is the resulting phenotype of the offspring after crossing? So red rose and pink rose. 50-50. Okay, problem number 2. A woman with a blood type O and a man with a blood type AB have expecting a child. What is the possible blood type of the kid? So we're going to identify the genotypic percentage as well as the phenotypic or the phenotypes. Okay, using key, I raised to A and I raised to B will result to AB blood, and small I or IO will result to double I will result to blood type O. So using Punnett square, small letter I times I raised to A will yield to I raised to A small I. Small letter I times IB will raise to I raised to B with small letter I. Same process. I raised to A times I will yield to IA small letter I, and IB times small letter I will yield to IB and I. So same process, if we see on the table, the genotypic percentage are 50%. So 50% blood type A and 50% blood type B. So if a woman with blood type O marries a man with blood type AB, so they are expecting a child with the probability of 50% blood type A and 50% blood type B. Okay, problem number 3. Determine the possible traits of the chicken if a block is mated with a white chicken. So write its genotypic percentage and phenotype. Okay, same process. Same process using Punnett square. A black chicken mated to white chicken. So B times W, VW, B times W, BW, B times W, BW, and B times W, BW. So 100% it will yield to checkered chicken. 100%. The possible offspring of this mating is checkered chicken. Okay, when we summary the 3 concepts using this table, incomplete dominance, when we say incomplete dominance, one allele is not dominant with one another. That's why it is called incomplete. When we say dominance, both alleles are simultaneously expressed on the heterozygote state of that organism. While multiple alleles, the gene is controlled by more than two alleles. Okay, given the entire example above, what is the implication of this? So what is the implication of this, class? It only implies that the characteristic of an organism indicates what kind of characteristic are they inherit from their parents. Next question. What is the advantage of having multiple alleles? So having multiple alleles class, it only make an organism more diverse. So if the organism are diverse, they have the capability to survive on the wilderness. So mas higher ang chances of survival. Next, if the characteristic of an organism is so diverse, what is the effect of this? So like what I said, the chances of survival are higher. Okay. In your own, using all the things that you have learned from our discussion, you're going to answer the following question. And then after that, you're going to email me your answer. Okay, I will read the question. Number 1, complete the Punnett square for a cross between a homozygous red-flowered snapdragon and a homozygous white-flowered snapdragon. Give the genotype, percentage, as well as its phenotype. Number 2, you're going to complete the table. It is already given the possible blood type. You're going to identify the possible gene pair. Okay. Number 3 question. Give one example of an organism and explain specific traits that make it unique to other organisms. Explain briefly. Okay, in your assignment, you're going to answer the following question. Number 1, define sex-related inheritance. Number 2, list down examples of sex-limited traits, sex-influenced traits, and sex-linked traits. I hope that our discussion are clear to you. If you have questions and queries, you can approach me. Just message me on my messenger or email me on my email. So I hope you learned from my discussion this afternoon, and I think you are all aware, or it awakens your mind why certain organisms are like that and like this. So you keep on wondering why that happened, or why. But because of this, it broadened your mind to the possibility of things. Thank you very much, and happy learning again.